All right, so last time we were talking about Mendelian genetics. So Mendel was studying genes that had a complete dominant and a recessive, and ones that sorted independently because they happen to be on different chromosomes. And we still today refer to those sorts of traits as Mendelian traits. So if we say a human trait is a Mendelian trait, and we'll talk about a few of those as examples, that means that there's a dominant and recessive gene, or a dominant and recessive allele, and the dominant is completely dominant. We don't see any in between the two phenotypes. But as everything in biology, it's never that simple. So we have other traits, which in fact is a lot of other traits that aren't Mendelian traits. So a trait could be controlled by multiple genes. That would be a non-Mendelian trait. We call those polygenic traits. We'll talk about that. Sometimes the dominant and recessive alleles are not completely dominant or recessive, and in that case, we can tell whether an individual has two copies of a dominant trait or one dominant and one recessive allele. Sometimes in fact, most of the time, genes have more than just two alleles. And in that case, that would also be non-Mendelian. And sometimes the same exact allele combination can produce different phenotypes. And there's, in fact, for many traits, there's a phenotypic range that we see. So Mendel did not pick traits that like any of these. So he deliberately picked traits that had a complete dominant and recessive allele for his studies, but not everything is like that. So we call those non-Mendelian. Non-Mendelian traits. So a trait is something we see in a phenotype. And there are quite a few traits that have many genes in different locations on different chromosomes that control that one trait. And height is one of those. Height is a good example of a polygenic trait. This is a famous photo taken by a geneticist at either Harvard or Yale, I can't remember. During World War I, they were recruiting student soldiers during World War I, which they did on many college campuses. And what they did was line them up behind a sign that had their height in inches. So each sign here has a height, and they differ by 1 inch. And about 100 individuals here have lined up for this particular thing. And you can see, and here's the same information on an actual graph, we can see that it forms almost a bell curve. It's close to a bell curve. And that is what we see with polygenic traits. That's in fact one of the ways we tell that a trait is not a Mendelian trait, but in fact is a polygenic trait. So what would happen if this was a Mendelian trait? If height was controlled by one gene and there was two or several alleles for height, what we would see is, instead of a bell curve, what we would see is discrete heights. We would have, some people who were 5 feet tall, and then nobody in between, and then some people who were 5'6", and then nobody in between, and then a few people who were 6 feet tall. We would not see a bell curve distribution. So when we see that for any kind of a trait, that is a really good sign that that trait is in fact controlled by many genes and is polygenic. So don't confuse that with polyallelic. Polyallelic would mean one gene that has many alleles. Skin color is another one that is polygenic, and this is a good example of one, and so we're going to use this as an example, a simplified version of that. So they are assuming here for this graphic that human skin pigmentation is only controlled by 3 genes, which they call A, B, and C, and that each one of these genes only has 2 alleles, a light allele and a dark allele, and that neither one is dominant over the other one. So that individuals who have a dark allele and a light allele, their skin pigmentation will be somewhere in between, halfway in between. So if we make those assumptions, and these these are mostly sort of true, but there are in fact more than 3 genes that control human skin pigmentation, and there is more than 2 alleles for each one. But it is correct that the alleles don't show complete dominance. There's no completely dominant alleles. So what happens is, if we take 2 individuals, so here's our 2 parents here, and both parents are heterozygous at all 3 genes, A, B, and C. So both parents have 1 dark allele and 1 light allele for each of these 3 skin pigmentation genes. So they themselves would have a skin pigmentation that we might call medium. But because they're heterozygous at all 3 of these, what we in fact get is they could have children who are absolutely anywhere on the spectrum, because gametes produce, because remember, gametes only have 1 of each of the parents' copies of these alleles. So a gamete could have all 3 dark alleles, all 3 light alleles, or any combination of those. But every gamete is going to have 1 A allele, 1 B allele, and 1 C allele. And then when fertilization occurs, those will recombine. So if these are our parents, then they could have offspring that are anywhere on the entire spectrum. Now, how would you get individuals that are highly heterozygous like this? Well, in fact, there are a lot of individuals in the United States that are very heterozygous, specifically because we have people in this country who have emigrated here either voluntarily or involuntarily from all over the world. So we, in fact, have a lot of people in this country who are heterozygous at many of these skin pigmentation alleles. If we put this all on a Punnett square, and I know this is a really big Punnett square compared to the ones that we have been looking at already. So, but what this shows, just like any other Punnett square, is the possible gametes from each parent. So if we have 3 genes here, and they're heterozygous at each one, each parent here is going to produce 8 different possible gametes with different combinations of the light and dark alleles. Because remember, the gametes are only going to contain 1 of each gene's allele. So one of the A alleles, 1 of the B alleles, and 1 of the C alleles. And it could be either the light one or the dark one. And I know they're using capital A and lowercase a as if there's a dominant and recessive, but that's not the case. That's just for convenience. So if we have 8 possible different combinations in the sperm, 8 possible different combinations in the egg, then we can see why, if we put this out in a Punnett square, that we can get every possible combination. But what you notice is combinations that are also very heterozygous are going to be much more likely. So if we count the number of squares that are the same and add them, because remember, for a Punnett square, if 2 squares are the same, then you can add the probabilities. So we have 64 squares here. So if every one of these squares is equally likely, which it would be, then if 2 squares are the same, then we can add them and make it 2 out of 64, or 3 out of 64, or 4 out of 64. So that's what we've done down here at the bottom. So what we in fact find is that there's only a 1 out of 64 chance that offspring would get all of the light alleles, and only 1 out of a 64 chance that they would get all of the dark alleles. And in fact, we have 15 out of 64 here for almost 50-50 light and dark. And same here, 20 out of 64 and 15 out of 64. So if we add up these middle 3 here, we get 50 out of 64. So 50 out of 64 possible offspring would have a medium skin tone similar to the parents. But these other ones, although less likely, are still definitely a possibility. So that's why sometimes in families you can have siblings, full siblings, who have very different levels of skin pigmentation, or the same. It would work the same way for height. Height is also polygenic. So we have multiple genes with various alleles controlling height. So especially if the parents have a lot of variability in their alleles for height, the offspring can have very different heights. You can get full siblings in the same family that are have huge differences in their height because of this. Pleiotropic effects. Now, pleiotropic effects in genetics is sort of becoming old-fashioned now. I'm going to mention it specifically because I know some of you are going to go on and take an upper-level genetics course, and it's important to know a little bit about pleiotropic effects. So back in the days before anybody had any idea what the molecular mechanisms were behind genetic inheritance, before they knew that it was DNA and that one gene controlled one specific protein that was being made or one specific RNA that was being made, before they knew that, they realized that there were some traits that seemed to be controlled by the same gene. So although the traits seemed to be completely unrelated to them, they were caused by the same gene. And we now know, now that we can trace back to the molecular basis for these traits in many cases, it becomes obvious that these traits are in fact not unrelated at all. And one example of this will be in the case of cystic fibrosis, which we're going to use cystic fibrosis as an example later in the chapter again. So cystic fibrosis is a recessive genetic disorder. So that means that in order to have the condition cystic fibrosis, you have to have 2 copies of the recessive allele. And we now know since the 1990s what this gene is. So the cystic fibrosis gene is a membrane protein that transports chloride ions out of the cells. And it's present every place in the body where there is a mucous membrane. So it's in the lungs, and that's primarily where the symptoms occur in individuals with cystic fibrosis. They have very thick mucus, and so they're at high risk for pneumonia and other lung diseases. But there are some other conditions that they also have. They tend to get blocked ducts in the liver and pancreas. Because of that, they have trouble with fat digestion. They also have extremely salty sweat and have trouble with electrolyte balance. So, and all these now don't seem like pleiotropic effects anymore, now that we know what the molecular cause is of cystic fibrosis, the fact that this membrane transporter is defective. So individuals who have 2 copies of the cystic fibrosis allele, they do not make a correct version of this membrane transport protein. And so chloride ions do not get transported across the lung mucosa. And so sodium does not come along, and therefore water isn't drawn out to dilute the mucus, because mucus is just protein dissolved in water. The protein is secreted by the cells that line these tissues, and then the water comes along because of the chloride ion transport. So if you take away the chloride ion transport, you get much less water dissolving that protein mucus, and it becomes very thick, and it doesn't move correctly. The cilia that are present in the lungs cannot flush out that mucus, so individuals who have cystic fibrosis have to cough constantly to get that thick mucus out of their lungs. So that's why I say pleiotropic effects are starting to be, this term is starting to become old-fashioned. Although if you're going on to another genetics course, you'll certainly talk about it. So pleiotropic effects is when we know that a single gene or a single allele is seemingly having unrelated effects on many systems in the body. So in cystic fibrosis, these things before the discovery of the gene seemed completely unrelated. Why should a person have problems with their lungs and their digestion and have salty sweat? Those just didn't make any sense. But now that we know what the gene is, now it makes more sense. Another interesting genetic disorder that we'll put that under the category of pleiotropic effects that, again, makes perfect sense once the gene was discovered, and we now know what it is. There's a condition called primary ciliary dyskinesia. And if we look at this, so ciliary, that means it has to do with the cilia. And we have cilia in many places in our body, including the lungs and women in the fallopian tubes. That's how the egg gets from the ovary to the uterus, is by being wafted along the fallopian tube by these cilia. And cilia and flagella are the same, just difference in length. So that means that the sperm swimming is by the flagella, which is a long cilia. They're exactly the same. So in individuals, so this disorder again is an autosomal recessive disorder. So autosomal meaning it's on one of the 22 autosomal chromosomes. Recessive meaning that it doesn't show up unless you have 2 copies of the recessive allele. So, and it is extremely rare, as things that cause infertility tend to be. Because remember, natural selection is about traits that improve reproduction. So any trait that removes an individual's chance of reproducing generally is going to be rare. So this is primary ciliary dyskinesia is rare. And so it affects places that you would expect. Respiratory tract is the cilia. So let's talk about what it is. So ciliary dyskinesia. So dys means not, and kinesis, that's, we see that root in there, like kinetic energy, that means movement. So dyskinesia means the cilia don't move. The flagella on sperm don't move. So they're there, we can see them, but they don't move. So these individuals get respiratory infections, sinus infections. Females are infertile because there's no ciliary movement to waft the eggs from the ovary to the uterus. There are also cilia in the ear, so those are affected. Sperm motility is non-existent, so males and females with this disorder are infertile. So, but a weird symptom or a weird part of this condition that was not understood until more recently is individuals with this also tend to have cetus inversus. So cetus inversus is either a complete reversal of the symmetry of the internal organs. So we look symmetrical, bilaterally symmetrical on the outside, but we are not symmetrical on the inside. Our heart leans to the left, and your liver is on the right, and your stomach is on the left, and the colon has a particular way that it curves around. And in almost everybody, it's the same. But there are rare individuals who either it's flipped, it's a mirror image, or their organs just look like a random jumble. So when a surgeon opens them up, it looks like somebody pulled out all their organs and just stuffed them back in kind of randomly. And often these people seem completely normal, and this cetus inversus is only discovered accidentally when they have an X-ray or something. So why should these go together? Why should individuals who have immotile cilia also have cetus inversus? That was weird and didn't make any sense, which is why it was placed under the category of pleiotropic effects. But now we know there are also cilia inside human embryos. So there's an early stage in embryonic development when all of us were a hollow ball of cells. And inside that hollow ball were cilia. And the cilia were wafting growth factors from one side to the other, and that's how we get our left-right asymmetry, is from those cilia in that very, very early embryo. So, pleiotropic effect of having cilia being immotile and also having cetus inversus, which seemed unrelated, but now we know it in fact has the same cause. So pleiotropic effects are usually explained once the molecular mechanism of a particular protein that's made by this defective gene is revealed. Another type of non-Mendelian trait would be incomplete dominance. Now, we already talked about the skin pigmentation genes which fall into this category. So I mentioned that if an individual has one dark allele and one light allele, that their skin pigmentation would be somewhere in between. That's what we call incomplete dominance. So one allele is not completely dominant over any of the others, and instead what that means is we can tell who the heterozygotes are. So here we have a gene for red flower color and a gene for white flower, or allele for white flower color, allele for red flower color, and the individuals who have one of each are lighter in color. So we can tell who the heterozygotes are, and that is what we mean by incomplete dominance. So if we can tell who the heterozygotes are because their trait is different than the homozygous dominant, then that's incomplete dominance. So we see this with a lot of human traits, like things like hair texture. So if one parent has really straight hair and the other parent has curly or very wavy hair, often the children will have some version of wave in their hair. So that's incomplete dominance. Dominance and how recessive and dominant alleles affect each other. Mendel thought that the dominant allele was in fact doing something to the recessive allele, covering it up, not changing it or diluting it, but stopping it from being expressed. And now we know that alleles in fact don't interact. So generally, if there's a complete dominant and recessive allele, the recessive allele is a non-functional allele. We now know that two alleles, they're on different chromosomes. They are just a DNA sequence that codes for something, either a protein or an RNA, or possibly a control region of a gene. And we can generally find different levels of interaction depending on what level we look at. So let's look at an example of that, Tay-Sachs disease. And Tay-Sachs is another one that we'll talk about later in this chapter as an example. So Tay-Sachs, like cystic fibrosis, is a recessive genetic disorder. So that means that in order to have the disorder, you have to have two copies of the recessive allele. Tay-Sachs is fatal in childhood. Many individuals with Tay-Sachs do not make it to their first birthday. Hardly ever do they make it to their fifth birthday. So it's an unfortunate disease with no good treatment. Individuals who have this, if the gene itself codes for an enzyme that helps get lipids out of the brain, so if you have a defective, two defective versions of this enzyme, in fact, you get an accumulation of fats in the brain that is harmful. But if we look at this gene and its expression at different levels, so if we look at the organismal level, so if we look at whole individuals, it appears that it is completely recessive. The Tay-Sachs disease allele is completely recessive because individuals who have one good copy of the enzyme gene and have one Tay-Sachs allele, so they only have one functional copy of that gene, they appear to be completely normal. So that's what we mean by dominant. So the normal allele appears to be completely dominant over the Tay-Sachs allele. But if we look at how this gene is actually expressed, so if we do brain biopsies and ask how much of this enzyme is actually present in people's brains, individuals who are heterozygous, that have one normal allele and one Tay-Sachs allele, they actually only have half of the amount of enzyme that we would expect to find. So at that level, that looks like an incomplete dominant because we can tell who the heterozygotes are. Remember, if it's an incomplete dominance, we can tell who the heterozygotes are because they're different. So at that level, now it looks like it's incompletely dominant. At the molecular level, it looks like it's codominant because in fact both alleles are transcribed as genes. So we'll talk more about what codominant is later. So if we look at, for Tay-Sachs, this graph just shows the percent of normal enzyme function in individuals' brains. So individuals who are homozygous for the Tay-Sachs allele, that is not a correctly functioning enzyme, and they have very little of this enzyme function. Individuals who are homozygous for the normal enzyme allele, they have two copies of the normal enzyme allele, so they have 100% of the activity. Heterozygotes, which we would call carriers for a recessive genetic disorder, they have 50% of the expected activity. So even though at the whole organism level, it looks like the normal allele looks like it's completely dominant over the Tay-Sachs allele, in fact, there is a difference. And this is going to become important for some of the other alleles that are genetic disorders, recessive genetic disorders that are weirdly not rare. So recessive alleles, the reason they are recessive is often because they are what we call null alleles. Remember that word null means zero. So that means zero good gene product is being made. Whatever that gene codes for, they are not making it. And that could be because the gene, the protein that's being made is defective. It could be because the gene itself is completely deleted. It could be because the gene is there, but the control region of that gene is mutated. And so even though the gene is there, it doesn't get transcribed. Any of those could cause a null allele. And we can't tell just by looking at a recessive phenotype which one of these it is. We would have to do more specific, especially DNA sequencing, to try to figure out which one it is. So it's just examples of two recessive phenotypes. So curly kale, which is very popular, flat leaf kale is the ancestral and dominant condition. Curly kale is a recent recessive mutation that has been cultivated because people like it. Here is an albino dolphin. This is kind of a famous one who lives off the coast of Hong Kong. And as of last year, he was still around. You could take tour boats to go see him because his family kind of hung around in the same place all the time. So albinism is another example of a recessive condition where an individual would have to have two copies of the same defective allele in order to have that condition. Now, dominant alleles are usually just the normal functional allele, like for the cystic fibrosis gene and for the Tay-Sachs gene. The dominant allele is just the normal functional gene, or the normal functional allele. But dominant alleles can also be deleterious, although this is less common, because what it means is if we have a dominant allele that is a deleterious allele, so here's another kind of old-fashioned word that's gotten frozen in biology, and we especially use this in genetics, and that's the word deleterious. So deleterious means it's bad. So a deleterious allele is an allele that is a harmful allele. So dominant alleles can be very rare, and polydactyly is an example of a dominant genetic condition that is still very rare. So what it does tell us, though, is that a dominant allele is not a null allele. That gene product is mutated. It's not normal, but it's definitely making something. There is definitely a protein being transcribed by that gene, because if it was, that's the only way we can have a dominant disease-causing allele, is it's not a null allele. It's making a gene product. So polydactyly is extra fingers and toes. Those are your dactyls. Usually it's extra pinky. Sometimes people have one or two extra pinkies. Sometimes they're really small compared to the normal fingers. This individual has an extra baby toe, perhaps also an extra pinky. Often these are surgically removed very young, and this is dominant. So we will look at that, the difference in how these traits are inherited. So just like Mendel's dominant traits, a dominant allele cannot hide. So if a child has one of these dominant genetic conditions, one of the parents also has it because it's dominant, unless there's been a new mutation, which does occur, but that's less likely. So if the child is born with polydactyly, very likely one of the parents has it, not as a carrier, because you can't be a carrier for a dominant allele. It's always expressed. We always see it in the phenotype. But, you know, like I said, it's often just the normal allele. So brown eyes is the dominant eye color, and it used to be the only eye color in humans. Blue eyes and green eyes are due to a recent genetic mutation in the last 10,000 years, and before that, for almost all of human history, everybody had brown eyes. That's just the normal functional allele. Codominance is often confused with incomplete dominance. Codominance, it's really hard to find good examples of codominance other than the A and B blood type, which we'll talk more about. But for codominance, both alleles are expressed. So I mentioned talking about Tay-Sachs, that if you actually look on the genetic level with Tay-Sachs, that you get both alleles, the Tay-Sachs allele and the normal functional allele for the enzyme, are both expressed. So, but if we can see them both in the phenotype, then we call it codominant. So for A and B blood types, that we call that codominant, because if you have one A allele and one B allele, your blood type is AB. We can see both in the phenotype. It's hard to find examples. Now, the examples that are used commonly in textbooks, these are two examples that are common in textbooks. One is, here's a flower that has white and pink petals being expressed. This is in fact not due to two different alleles. This plant is not heterozygous for white and pink flower color. Usually this is due to a jumping gene, as they used to be called, or possibly even a virus that is inactivating a gene in some of the clonal lineages in the development of the flower, but it is not due to codominance. Another example that's commonly used, this is a horse that's got a coat color that's called roan, R-O-A-N, and if you look really closely at the hairs, the individual hairs on the horse, they have white hairs and black hairs, and together they make this kind of silvery blue look. Although I have been informed by former students who were horse breeders that in fact you do not get a roan horse by breeding a white horse and a black horse and then the offspring are a roan horse. So it is not due to, this color trait is not due to the codominance of a white allele and a black allele. So it's not really a very good example. It is extremely hard to find decent examples of codominance. So the best one that we have is the A and B blood groups, which we'll talk about. So for almost all genes, there are in fact more than two alleles, multiple alleles. And when Mendel did his experiments, he deliberately narrowed it down to two for the genes that he studied. But in fact, even the genes that Mendel studied, if you look at all of the pea populations that we have, the different seed varieties that are available, in fact, you could find more than two. But let's use our blood type groups as an example. So we'll talk about the positive and negative part of blood type as well. So for blood type, you either have A, B, O, or AB. Those are the possible blood types. And we get those, there's one gene that codes for an enzyme, and that's why we use the letter I here to represent the allele. So the capital I is used for the A and the B allele, and a lowercase I is used for the O allele because O is a null allele. So this gene, or this particular allele, codes for a defective enzyme. It's not functional. So so A and B are both dominant over O because O is a null allele. So what they do though, what this enzyme actually does, that starts with an I, we won't worry about what the name of it is, but that's why we use I for the alleles. There are proteins sticking out of the surface of your red blood cells. And some of those proteins can have carbohydrates attached to them. And this enzyme, that's what it does. It adds carbohydrates to this particular protein that's sticking out of our red blood cells. And it can add, if you have the A allele, it will add an A-type carbohydrate. If you have a B allele, it will add a B-type carbohydrate. If you have an O allele, the enzyme doesn't function and you get no added carbohydrate on the surface of your red blood cells. So this is going to affect how your immune system responds to your own blood. It's not going to respond to your own blood. And if you get a blood transfusion, other people's blood, if it ends up in your body. So this Punnett square doesn't represent the reproduction of two individuals, but it does represent how we get all the different blood types. So if you look at this, if you have two A alleles, or an A and an O allele, remember this is the null allele that we call the O allele, then you're going to be type A. So two different possible genotypes for blood type A. If your blood type is B, then you could be homozygous for the B allele, or you could have one B allele and one O allele, because you remember A and B are both dominant over O, because O is a null allele. If you have an A and a B allele, then your blood type is AB. If your blood type is O, that means that you're homozygous for this null allele, for the O allele. So there are two blood types here for which if you have that blood type, we know your genotype. If your blood type is AB, we know your genotype. There's only one possibility. You have to be heterozygous and have one A and one B allele. If your blood type is O as well, we know your genotype because there's only one possibility. You have to be homozygous for the O allele. You might think that type O would be the rarest blood type because you have to be homozygous, and that these other blood types would be the most common, and that A and B might be equally common, but that's not at all what we see. In fact, O and A are the most common blood types, and B is more rare, and so is AB. And it depends a bit where you are in the world. The B allele is the most common in the Middle East and becomes more rare the further away from the Middle East you get. And among Native Americans, the B allele was non-existent until the first Europeans showed up with their B alleles. So in fact, A, B, and B are rarer types. Now this is different from the Rh factor, which we'll talk a little bit about. So the Rh factor, which is the plus or the minus in your blood type, that is a different gene, a different chromosome, but it's similar. It's an enzyme that adds something to the surface of your red blood cell. And if you are plus, if you are Rh plus, that means that you have the enzyme. And if you're negative, that means that negative is, again, is a null allele. That means that you are missing a good copy of that particular enzyme. And again, just like O, that means you're homozygous. So if your blood type is O negative, that means that you're homozygous for the O allele, and you're also homozygous for the Rh negative allele as well. And this is, again, this is a simplified picture of blood type. Like everything in biology, there's always more levels of complexity here. And there are, for the A allele, there's like a common A allele and more rare ones, and the same for the B. And even for the plus and minus Rh factors, there's also more rare alleles that go along with that as well. So probably you know that certain blood types can donate to other blood types, and it depends on the recipient's blood type. And the reason is because of something called antibodies in our blood. So anything that your immune system might react to is an antigen. So things on the surface of a pollen granule are antigens. Proteins on the surface of bacteria or viruses are antigens. They're things that your body's immune system might react to. Now, these carbohydrates on the surface of our red blood cells that the A and the B enzyme are making, these are also antigens, but you don't react to your own. So we call that self. So you will not have antibodies against self. And so if that means if you're type A, you will have anti-B antibodies, but not anti-A antibodies. So antibodies are made by cells in our immune system, particular white blood cells, that react to specific surface proteins on bacteria and viruses, hopefully, but also against cancer cells that are making mutant versions of our own proteins that are now not recognized as self. So, but if somebody is blood type A and I give you blood that has B antigens in it, your body will react, and it will react by these antibodies will clomp onto those B antigens, and it will cause clots to form in your blood. And as you can imagine, that is a very dangerous and life-threatening reaction, that kind of cross reaction. So generally when they're giving blood to people, even if they think they're giving you a compatible type that you won't react to, they usually do a little test mix and mix a little bit of the donor blood with the patient's blood just to make sure that it's not going to clot, because just being the right type doesn't necessarily mean that there won't be a reaction of some kind. So if your blood type is AB, that means you don't have antibodies to either A or B. If your blood type is O, you're going to react to both A and B. So what that means is that if your blood type is AB, you can get blood from A, B, or O, because you're not going to react to the A and the B antigens. If your blood type is O, however, you can only get other O blood. And the same is true for the Rh factor, the plus and the minus in Rh factor. So if your blood type is AB positive, that means you could get blood from anybody, because you're not going to have antibodies against the A, against the B, or against the Rh plus. If your blood type is O negative, you're going to have antibodies to all of them. So you can only get blood from other people who are O negative, but that makes O negative people the universal donor, because type O's, they don't have any of those antigens. They don't have the A or the B antigens. If you're O negative, you don't have the Rh positive antigens either. So that means we can give your blood to anybody and they won't react to it, unless they have one of the rare subtypes. So blood type AB can receive blood from anybody, but can only donate to other type ABs. Type O, you can donate to anybody, but you can only receive from other type O's. Now the opposite is true for plasma donation. We've been hearing a lot about blood plasma donation lately as it relates to the COVID-19 crisis, and that if people who are recovered from COVID-19 could possibly donate their plasma, and that could be used to treat people who are very ill. Well, as it happens, if you're donating plasma and your blood type is AB positive, because we're taking all the cells out, we don't give anybody the cells when we're donating plasma. What we're donating is the liquid portion of the blood. So somebody who's AB positive, you're not going to have antibodies against any other blood type. And so for plasma donation, AB positive is the most desirable. For blood donation, O negative, and for whole blood donation, O negative is the most desirable. It's the opposite though for plasma donation. So if you're type AB, you're probably better off donating plasma. If your type is O or A is the other really common type. If your blood type is O or A, donate whole blood, because your blood is needed. So another way that traits don't behave like Mendelian traits is when the environment plays a part in how the phenotype is decided. So some things, there's very little what we would call nurture or the environmental influence on a trait. Like, what is your eye color? Well, there is very little environmental influence on that. But other things like your height, there is a phenotypic range. For example, if you don't receive adequate food as a child, your height will be shorter than if you did receive adequate food as a child. And we would call that the phenotypic range for a particular genotype. So whatever the genotype is, there is a phenotypic range, especially for polygenic traits. And even for some of these recessive genetic disorders that we will talk about, there is a phenotypic range. So for example, for Tay-Sachs disease, some individuals don't make it to their 6-month birthday, but some other individuals make it until they're 4 or 5 years old. So even though they have the same recessive genetic disorder, there is a range for that. Even for things like Down syndrome, there's a phenotypic range. And we'll talk more about Down syndrome and when we get to the next section. But some individuals, they all have the same thing. They have an extra chromosome 21, but some individuals are more severely affected. Some individuals never speak, can't learn to take care of themselves at all, or dress themselves, or feed themselves. And other individuals who have the same thing, who have Down syndrome, can hold a job and can live somewhat independently with a little bit of help. So there is a big phenotypic range for surprisingly a lot of genetic conditions. So here's one example. So from plants, so hydrangeas are a really popular kind of plant. I have hydrangeas, and the blue hydrangeas are very popular because blue flowers are kind of unusual. So gardeners like blue. But the phenotype will change depending on what type of soil you have. And so the state I live in, we have pretty alkaline soil here. It has a lot of clay in it, and that is not very good for the blue color in hydrangeas. So I had some of these blue hydrangeas, and the first year they were blue, the next year they were kind of purple, and after that they've been pink. And that's because of the pH of the soil, because they need a more acidic soil, but they also need aluminum in the soil. This blue color, the pigment itself, uses aluminum. So there has to be aluminum in the soil that the acid pH helps the roots to get access to. But the genotype doesn't change. So whether the flowers are pink or whether they're blue, the genotype is the same. It depends on the environment. So that's an example of phenotypic range. So for this one genotype, we can have different flower colors depending on what the environment is. Now, sometimes the phenotype will vary within one individual. So probably you've all seen a Siamese cat. There's another related cat variety called a Himalayan that sort of looks like a Siamese cat, but it has longer hair. And they have a defective enzyme. Now, there are several enzymes that make the dark pigment melanin. And for Siamese cats and for Himalayan cats, there's a mutation in one of these enzymes that makes it not function at normal cat body temperature. So cats' body temperature is a little bit warmer than humans. It's about 101, 102 Fahrenheit. And this enzyme does not function at that temperature. So we don't get melanin in places in the body where the cat's skin is warm. Instead, it functions at a cooler temperature. So places on the cat's body that are cooler, like especially the ears and the tail and their paws, we get darker pigment because the melanin producing enzyme can now be active at slightly cooler temperatures. So every cell in the cat's body, though, has the same genotype. It's just that in some places in the body, that particular enzyme is not functioning because it's warm. Now, you can affect the color of the cat's fur by warming up parts of the body. So cats that have had surgery or something, and they have to have their paw wrapped up with medical tape or something, that's going to keep it warm. And when the fur starts to grow in fact, it will be lighter in color because that enzyme will now be non-functional in the part of the body that's being kept warm. So genotype is the same, temperature is different, so that changes the phenotype. Now, the phenotypic range is especially broad for polygenic traits. So remember, polygenic, that means that multiple genes control that trait. And so if that's the case, if we have a broad phenotypic range that's due to environmental considerations, we call those traits multifactorial, meaning that there's a genetic component and there's an environmental component to that trait. And collectively, those go together with the phenotype. So something like height, Also IQ are very affected by environment as well as genetics. So for height, if you don't receive enough food as a child, you will be shorter than you would have been otherwise. For IQ as well, IQ is increased with schooling, education. So IQ is going to be affected by environment as well as genetics. And as you can imagine, sports performance and grades in school also are not just due to innate genetic ability, but to environment as well. So those would be examples of multifactorial traits. If the condition that we're talking about, the trait that we're talking about, is a disease or a disorder, then we would call that multifactorial. And many of the current diseases of concern, things that are the so-called diseases of civilization, are such multifactorial conditions like heart disease, diabetes, alcoholism, mental illnesses like schizophrenia, and cancers have both a genetic component and an environmental component. And we can often see that when we look at families. We can see, oh yes, there's several members of the same family who have heart disease or who died of a heart attack below age 60. So that tells us there's a genetic component, but there's also a strong environmental component, and especially if a disease prevalence has changed recently, like let's look at type 2 diabetes. So type 2 diabetes 100 years ago was an extremely rare condition. Now it's very common. Type 2 diabetes has gone in my lifetime from being called adult onset diabetes because only adults had it, to being something that also affects children. Starting in the 1990s, children started to be diagnosed with type 2 diabetes. So that's a change. And when a disease prevalence changes rapidly like that in a matter of just a couple of generations, that is not genetic change. That is due to an environmental change. The environment has changed, not the genetics. But it is absolutely true that some people have a higher likelihood in the same environment of developing type 2 diabetes. So there is a genetic component, there's also an environmental component.